So as chair of the Federal Committee of the Plant Conservation Alliance, I'm pleased to welcome everyone to this meeting and to introduce our speaker, Ms. Sarah Fitzsimmons, Chief Conservation Officer for the American Chestnut Foundation. Sarah has worked at Penn State University with the American Chestnut Foundation since 2003, assisting chestnut growers and researchers throughout the Appalachian Mountains. She serves as the Regional Science Coordinator for the North Central region, which includes the state chapters of TACF in Ohio, Pennsylvania, New Jersey, New York, and Indiana, and hopes her research and professional work will facilitate long-term conservation and restoration of native tree species at risk from exotic pests and diseases. Please join me in welcoming Sarah Fitzsimmons talking to us today about restoring the American chestnut for robust forests in the eastern U.S. Thank you.
Excellent. Thanks so much. And thank you all for inviting me to speak with you all today. So this is one of my favorite pictures. This is a wild American chestnut on top of a mountain in Western North Carolina outside of Asheville. I unfortunately did not take this picture. I can't take credit for it, but I really love using this to introduce kind of the vision of what we want to see, which is chestnuts in the mountains of the Appalachians across the eastern U.S. and producing lots of fruit and mast for the critters of that ecosystem. As mentioned in my bio, I'm really hoping that the work that we're doing with the American Chestnut Foundation and many of our partners can help overcome what seems to be, unfortunately, an unending invasion of non-native pests into the U.S. You can see from a couple of these charts there on the left, you can see that chart with everything going up. That's a number of non-native forest insects and over time that have been introduced into the United States. On the right is a scary map of the number of pests per county, and you can see how under threat the northeast part of the U.S. is in particular, and of course the West Coast, ever expanding due to mostly expansion of global trade. And I think this headline is a little hyperbolic, but really portends what is in our future with how many tree species are under attack due to threatened pests or threatening and invasive pests and diseases, but also vegetative pests and climate change as that shifts their ranges into unknown territory. So this title here from the Washington Post: as many as 1 in 6 U.S. tree species is threatened with extinction. And again, that might be a little hyperbolic, but we can see it all around us. I'm in central Pennsylvania. I work out of Penn State, and 2 of these pests, hemlock woolly adelgid, emerald ash borer, are already here, causing lots of mortality, sudden oak death. I just hope that I will never see that in person in the eastern U.S., and hopefully we can do things to reduce its impact in the West Coast. But the work that we're doing with the Chestnut Foundation, some of the technological advancements we've been able to make with our partners, and also some of the work we're doing with citizen scientists and just the kind of template we've set with partnerships, I'm hoping that this can lay the groundwork for rescue and reintroduction of these tree species that are already affected. So this is sort of a case study of the American chestnut and what we're doing, what happened to the American chestnut and what we're doing to bring it back. For those of you who are not familiar with the story of what happened to the American chestnut, it at one point was a widespread species in the eastern U.S. Its range is from Maine to Georgia, west into Indiana and Michigan, primarily the Appalachian Mountains. And in the late 1800s, when the nursery trade, especially the exotic nursery trade, was really in full swing, Japanese chestnuts were being imported into the U.S. And on those Japanese chestnuts hitchhiked a fungal disease now called chestnut blight fungus. And you can see the trees on the left. This is a haunting photograph from Shenandoah National Park, where you can see just massive amounts of chestnut trees dead from the chestnut blight. And we're seeing similar Unfortunately, similar scenes in play in areas where, say, ash trees are extensively growing. On the right, you can see what we see today. Most of the American chestnut trees that we see in the landscape at this point look like the tree on the right, which is a blighted stem in the middle, surrounded by lots of sprouts surrounding it, which then get infected by the blight. And here's a sort of a backing up and looking a little bit farther of one of these trees. You can see all these gray stems here in the middle. These are all dead stems of American chestnuts that are then replaced by sprouts around it. There are some 430 million American chestnuts still on the landscape, but they pretty much, 84% of them look like this. 84% are less than an inch in diameter. But because there are so many, because there are an estimated 430 million trees out there, it's not threatened, it's not endangered, it's not certainly not extinct. But we call it functionally extinct because as a little shrubby, sprouty species, this tree is no longer performing its ecological services in the ecosystem. Historically, the tree was used extensively for various products. I've heard it called a cradle-to-grave tree because it was used for cribs, coffins, and everything in between. I love this picture in the lower right. Hot chestnuts, good for the brain. Try a bag. They are pretty tasty. Chestnuts roasting on a fire, open fire, obviously, I think is how most people are familiar with them. I grew up in southern West Virginia, and I'm really familiar with the split rail fences, some of which still exist today from American chestnut. This is another picture from the Shenandoah National Park, just showing how widespread the wood was in use. And then, of course, I don't think it's any secret to this group of how important native tree species are to the food web. A lot of groups I speak to are really surprised to see things like caterpillars or fish on this photograph, but obviously the native tree species and the amount of insects that they feed is known to be so important at this time, more than ever. We understand that better than we ever have in the past. And leaf litter, for example, will then fall down, decay, feed aquatic larvae, which then feed fish. So native tree species and their importance to the entirety of the food web can't be understated. And American chestnut plays a huge role, or once played a huge role, and can again there. I think one of the most noticeable aspects of chestnuts are the nuts. So you can see here on the right, upper right hand, American chestnut burr and nuts. So there is a burr that develops from the flower. So here's the flower beside it. She looks like a little green pineapple. This picture is taken probably around July 4th. That's when chestnuts are at the height of their blooming. This is a bisexual catkin. So the male flower is a catkin, which look like long, fuzzy yellow pipe cleaners. And then at the base of some of the catkins are a female flower. That female flower, once it gets pollinated, will produce or can produce 3 nuts in a burr. The burr looks like a sea urchin, so we sometimes call them tree urchins. They're very pointy, very spiky, to protect the developing nuts until they're mature so that they can then be dispersed by things like blue jays and squirrels and everything else that loves to eat hard mast. And so you can see all these really pretty chestnuts there behind it. One of the things about the decline of the chestnut and what happened was throughout a majority of its range, oak took over the niche that chestnut played in the ecosystem. And while oaks have they're in the same family, they're all in the Fagaceae, and while oaks have a very similar nutritional profile, or acorns have a very similar nutritional profile to chestnuts, typically chestnuts can produce more seed earlier. They can produce seed more regularly. They tend to produce a regular annual crop versus the cyclical nature of acorn production. And they fruit earlier than oaks, and they tend to produce more mast per unit area. So there are some estimates that say that we have not been able to recover upwards of 40% of the mass production that American chestnut provided to the ecosystem, that oaks have not been able to make up some 40% of what chestnuts were able to produce in the eastern U.S. forests. So what do we do about this? How do we bring the chestnut back, or any other species back? And so it's as easy as 1, 2, 3. The first thing you got to do is go out there and find the remnant germplasm. Then you have to identify one or more sources of resistance or control. You put those together and you put them back out in the woods. And so while the steps themselves sound pretty simple, as always, the devil's in the details. I'd say number one is relatively the easiest, as long as you're dealing with a species that isn't being threatened with extinction. Finding the sources of resistance or control are going to be the biggest issue. That's the kinds of things we're dealing with with ash right now, as an example. And then getting back out into the woods, right now, I don't know that we have a really great example of how to do that with hardwoods. Longleaf pine shows a really great example of how habitat restoration has allowed for an amazing increase in that species' ability to thrive. So both habitat restoration and some reinvigorating more traditional methods of forest management that encourage longleaf pine regeneration. But hardwoods we're pretty in a very early stages of knowing how to properly artificially regenerate hardwood species. So that's going to be a challenge moving forward, one that we're excited to embark on. So I work with the American Chestnut Foundation and Penn State University. I'm an employee of both organizations, but all of my work is funded by the Chestnut Foundation, which began in 1983. There are over 5,000 members spread across 16 chapters. And what I love about the model of the American Chestnut Foundation is no singular organization can really take on, and as a group like this, the Plant Conservation Alliance, I think, can also well describe. No singular organization can restore a species, and especially a tree species. It takes the work and effort of all kinds of different people working on an amazing amount of different types of disciplines to solve these kinds of problems. And that's really where the American Chestnut Foundation is more of a hub of people and work to make sure that we're not duplicating effort and or that we're following priority initiatives that work toward species restoration. So let's go back to that stepwise work of, first you got to find and conserve that germplasm. So for trees, there are other tools out there. iNaturalist, I don't want to downplay the importance of apps and software like that. They are great. We prefer TreeSnap, which is a piece of software, an app that was created by the University of Kentucky, Tennessee, and then the Forest Service. This application is very specific to finding and tracking and documenting threatened tree species. And the reason we prefer it is because here on the right, you'll see, this is in the application on the phone app, we can take pictures like you can in iNaturalist, but it also gives us a lot of really important information. Is that tree producing nuts, burrs, or catkins? So is it flowering? Does it have the blight yet? How big is it? How tall is it? All of these things are really important for us to know how can we use that tree, especially if it's flowering and producing nuts. That's a much easier way for us to conserve that genotype, that provenance, than if it's not. So we're going to prioritize trees that are fruiting over ones that are not. And that location information is vital. Also captured by iNaturalist, but TreeSnap just gives us a little bit more demographic information that's important for us to prioritize. I mentioned earlier that there are some 430 million American chestnuts out on that landscape. And so here's a map of the current population. This is a map that was done by John Scrivani, who is retired from the Virginia Department of Forestry, currently an adjunct prof at University of Richmond. He looked at FIA data of American chestnut, and this is really close to what we find from data on iNaturalist and also TreeSnap of where our partners and citizen scientists are finding trees. So you'll see a huge population there in the in the mountains of western North Carolina, eastern Tennessee, up through the Shenandoah Valley, all kinds of trees in Connecticut, Massachusetts, up into southern Maine. Now there are populations elsewhere. You can see this disjunct population in northern Michigan, over here in Arkansas. What's really interesting about this is you see places where the range is historically noted, and if you read some of the historic literature, if you read historical publications from the Indiana Department of Agriculture, there are accounts of how numerous and big chestnuts were in southern Indiana, particularly Brown County. You'll read accounts in southeastern Ohio of how important chestnut was to the ecosystem there, and now virtually extirpated. It is very rare that we find trees in those locations. Same here in South Jersey. In the Pine Barrens. I've been scouting there for several years, and we have found a few. I mean, and when I say a few, like maybe a dozen, but it's very difficult to find them now. So we are unfortunately experiencing a range constriction, which is kind of expected for a species that is no longer regenerating and sexually reproducing. But there still is a wide variety, or are a wide variety of trees from which we can collect native germplasm and conserve. Here's the data that this is combined data from TreeSnap and iNaturalist, and also some other reports that we get from the American Chestnut Foundation. And you can see that kind of mimics what's in the FIA data from John. You can see some of these disjunct populations. There's Wisconsin, Michigan, but huge amounts of trees throughout Massachusetts and Connecticut, down through the mountains of western North Carolina, eastern Tennessee, couple blips there in Indiana. But you know, really wide swaths where the tree where that range is constricting. So how do we do anything about that? So this group, I'm sure, is more familiar than any other that I speak with about ex situ conservation and getting these seed or these genotypes and these lines conserved for diversification purposes of blight or disease-resistant material. Unfortunately, chestnut is one of those highly recalcitrant species. The seed don't store well. Viability drops significantly after 6 months. I have not been successful keeping seed for longer than 2 years. Vegetative propagation is very difficult. It doesn't readily root. There's delayed grafting compatibility and blight infection that affects grafts. Tissue culture is currently only really effective on embryos, although you can use some cyanwood, and rooting, unfortunately, it's what I call the holy grail. If we can figure out how to root chestnut or some of the other similar recalcitrant species, that would be really phenomenal for improving the success in a lot of the various activities that we do in germplasm conservation and breeding. But unfortunately, what the easiest way to conserve chestnut at this point is with a nut. If you're going to do that, you got to find them. Again, most of the trees are like this tree on the right, which are in the understory. They're blighted. If nuts are the easiest to propagate, then something like this guy here, this was found by a flyover from students at the University of Maine. Brian Roth was their mentor at the time. And he took them across. He first modeled suitable habitat at locations where it was expected they could find wild American chestnuts. They flew planes over. I'm not sure what amount of area they covered, but quite a bit of southern Maine, and they found what ended up being the tallest American chestnut in the state. If you do these flyovers at the height of flowering, you can see they really stick out like a proverbial sore thumb. They're very noticeable. And then you can go in and collect seed from them and put them in what we call germplasm conservation orchards, basically ex situ gene banks. And the main reason is for diversity. I like this picture. It's a little grainy and it doesn't show, I'm not sure how well it shows up on your all screens, but on the left is this is taken on the same day. These are from the same provenance, but you can see on the left, the tree on the left has hardly leafed out. The tree on the right is halfway leafed out, and that's the kind of diversity that you want to be able to both capture and also implement into a resistant population when you redeploy. You need some trees that are going to leaf out early, some trees that are going to leaf out late, so that they can respond to the various climatic triggers that will allow them to thrive with phenological response. So that's a big question for us. How much diversity do we need to conserve within the species and capture? Well, how much diversity, first of all, is in the species? How much of that do we then need to capture and reintroduce into disease-resistant materials? How much do we conserve in the wild versus how much we capture in gene banks, and in this case, primarily orchards or maybe frozen pollen? And do we have any control sites where we don't plant disease-resistant populations? So that all dealt with step number one, which is finding and conserving the remnant diversity. The next thing you have to do is find mechanisms of resistance. And right now for chestnut, there are three different ways, three categories of ways, and I'm going to highlight the major focuses of those three categories next. But we call that breeding, biotech, and biocontrol united for restoration, or 3BUR. And I love this. It's super corny, but it's really memorable because there are three nuts in that chestnut burr, and we've got three Bs that we focus on, that we mush together. No singular one of these techniques is a silver bullet. While any one might be better than another, they can't function on their own. So breeding is going to be necessary, biotech is going to be necessary, biocontrol is going to be necessary for different aspects of this project. Biocontrol probably has the least amount of impact based on how much it can control the fungus, but it certainly allows us to improve our conservation of wild type trees. On the left, you can see uninfected mature bark of an American chestnut. On the right, or in the middle, you see a non-lethal canker. So if you see this, what we call scruffy bark, this is either an indication that the fungus is sick from hypovirulence, that a virus has infected the fungus, or it could be an indication that the tree has some amount of resistance. It could also be a combination of the two. But hypovirulence is a phenomenon where the fungus gets infected by a virus. Over here on the right, you see all these plates. Anything with the white fluffiness is a fungus, is a chestnut blight fungus that is invaded by a virus. And there's a lot of intricacies that go into the utilization of this type of biocontrol. One of which is if you make your fungus too sick, then that virus will not spread across the population. So you can't have too strong of a virus. And if you don't make it sick enough, well, then it's not going to save the tree. So there's a lot of intricacies that go into hypovirulence. It is something that we still study with our partners at WVU, the University of Maryland, and some other locations. And like I said, it's going to be and will continue to be an important aspect of long-term conservation of American chestnut germplasm. Breeding is our second B. This is the Arboretum at Penn State. This is a breeding orchard that I've been working on there since I started in 2003. On the left, you can see we plant tens of thousands of trees. And in order to find those that have the right combination of disease resistance and growth. So on the left, all that pink paint on the bottom means that those trees don't have what it takes. So we go in, we cut them down, we throw them on the pile, and we have a weenie roast. On the right, you see the trees that we keep and then do additional breeding on. You can see the growth form is very characteristic of that tall upright growth, very straight, a straight bowl with acute angles of those branches going upward. This tree here that I'm standing next to with the catchy name of 32260 is one of the best trees that we have in this arboretum planting, and we've used extensively for breeding and deployment into the woods. Of particular interest right now is the biotech root, and this has been pioneered. Bill Powell there on the right holding the chestnut tree. He works at SUNY ESF, the State University of New York's College of Environmental Science and Forestry, and he has been in charge of the work of genetically engineering an American chestnut. The first job was to try and get some sort of tissue culture working, and he was able to do that with a colleague, Chuck Maynard, who's now retired. And that was where they worked with embryos, putting them into tissue culture. The next step was finding a gene that could then be inserted into the tree, and that gene was OXO. And that OXO gene stands for oxalic oxidase. It's a gene from wheat. And Bill actually had worked with hypovirulence previously. And there's some, what happens is the chestnut blight fungus secretes oxalic acid. It does that in order to kill the chestnut tissue ahead of it eating. And so because the chestnut blight fungus kills the tissue before it eats it, it's known as a necrotroph. It eats dead tissue. But that oxalic oxidase gene neutralizes the oxalic acid produced by the chestnut blight fungus. So then he worked on getting the gene into the plant through agrobacterium mediated transformation. That work took about 40 years. And I'm actually going to see, I don't know if I can put it in the chat while I'm presenting. I'll have to do it afterwards. We are actually in the middle of a public comment period right now with USDA APHIS. This is a public comment period on the EIS and PPRA, the Environmental Impact Statement, and Plant Pest Risk Assessment. It's the draft EIS and PPRA submitted by USD APHIS. As soon as we finish up here, I'll put a link in the chat so you guys can comment. It was originally a 45-day public comment period that was started in mid-November. Several organizations asked for an extension, and so now that public comment period has been extended to January 27th. So you have a couple weeks to still comment. I urge you guys, hopefully, to make a supportive comment. But it'll be the first example of a tree being of a genetically modified tree being used for wild restoration for forest health purposes. There have been 3 other types of trees deregulated through APHIS, the apple, plum, and papaya. There are 2 types of apple that have been released. Cold-tolerant eucalyptus has been in purgatory within this process for some time, and has not yet met the safety requirements that APHIS puts on these organisms. But what's really exciting about the draft environmental impact statement and plant pest risk assessment by APHIS is they have found through 2 years of study, well, 2+ years of study and other public comment periods, they see no risk of, and in fact, benefit to releasing the blight-resistant American chestnut, blight-tolerant, genetically modified American chestnut. I'll mention too that the name of that is Darling 58 or D58. So if you go, if you just Google Darling 58, you'll probably come up with lots of information on it, both from ESF and the American Chestnut Foundation. A little bit about the safety. What went into the petition to APHIS are a lot of studies looking at how do these transgenic trees act in comparison to their wild relatives. Does the and I'll go from left to right and top to bottom. Does the leaf litter decompose the same? It does. Do seedlings germinate in leaf litter the same in transgenic as wild type? And they do. Do caterpillars eat the leaves of transgenic American chestnut the same way they do wild type? They do. A bigger concern would be mycorrhizal fungi. If you have antifungal properties, are you still going to have mycorrhizal colonization? And you do. The bee health was the same, and tadpole development was the same. There are other studies that go into that position just to show the overall safety of this transgenic tree. So those are the 3 Ps. That'll wrap up step 2. So step 1 was finding and conserving your diversity. Step 2 was finding some source of resistance and getting them into that, getting them diversified with the population that you've conserved. Step 3 is getting them back out into the woods, and that's primarily what I work on in my day-to-day research. Is a lot of the getting trees back out in the woods. This is a colleague of mine, Paul Lupo, who now works with the Pennsylvania Game Commission, and Alex Harding, who was at Penn State for some time. This is on the left is an example of where I don't really want to plant chestnut trees. So a lot of people ask, well, where are you going to plant these things, and how much are you going to plant versus let wildlife take into the landscape? Well, this is classic Allegheny woodrat territory. Here in this picture is Greg Turner with the Game Commission. He and my colleague Hoy were out putting in supplemental feeding plots of chestnuts into these really rocky areas. Chestnuts love growing in the middle of rock piles. They love sand, they love rock piles, they grow really well, but I don't really want to plant there. I would much rather plant at the base of those rock piles where it's easier to plant, and then have the rats grab them and plant them up in those rock piles. So dispersal is going to play a huge role. I've had the pleasure of working up at a site in central Maine, in Atkinson, Maine, looking at dispersal of a naturally regenerating site and tracking the life cycle of trees. This is an example of a cache that was forgotten by some red squirrels. Red squirrels are the primary dispersal agent of the site up in central Maine. So we've been doing a lot of work trying to model how different types of wildlife are going to spread the species once you get sites installed. We also had a great colleague at Ohio State who just graduated looking at blue jay dispersal, and that's again entering into that model of figuring out, well, how many trees do we need to plant and how many places in order to get enough dispersal to get long-term regeneration and installation of blight-resistant, disease-resistant populations. Just another look at the site in Maine, how we've mapped all these out. We've mapped tens of thousands of trees in this site to try and get a sense of, and this year, we're working with the Hamilton Lab, the Schatt Center here at Penn State, to look at founder effect and inbreeding coefficients. This site in particular in Maine was founded by very few individuals. And we've got another example at West Salem, Wisconsin, Berlin, Vermont, Roselawn, Indiana, where you have hundreds, if not thousands, of interbreeding individuals that were all based on very small founder populations of anywhere from 4 to 9 individuals. And so we're going to look at some of the molecular underpinnings of that, which have great impact on us figuring out, well, how many genotypes do we need to impart on these restoration populations? We'll hopefully have those data in the next 2 years. So there's not only how many provenances or how many backgrounds or how many genotypes do you need to put in order for long-term restoration and population turnover, how many trees, period, do you need to put in there? We've been using some modeling that was done by Rogstad and Pelican in 2011 and 2014. There's a lot of words here. Basically, what their work working at the West Salem stand in Wisconsin, they found that in order to maximize allelic diversity across a stand, you should work with about 300 individuals in the beginning, which ends up with about 169 trees breeding a breeding population. And that's really what we see in our populations that we've planted with the American Chestnut Foundation. We see about 50% survival long term over decades. So this really holds up over our observations too, that whatever you plant, you can expect to get 50% or less of them surviving to reproductive age. So let's use that as the baseline of what we'll call a reintroduction plot. So we take 300 individuals of blight or disease-resistant individuals. We'll plant them in a location. How do you get that many seeds and seedlings? Here's some containerized trees we have at Penn State in our greenhouse. This is Bruce Wakeland there on the right, planting seed at the Valonia Nursery in Southern Indiana. And shout out to them, because that, despite compared to the Allegheny wood rat rocks, planting in Indiana is really a phenomenal experience. The soil is so deep and beautiful and easy to work. It's a nice comparison to the rocks that I typically plant in Pennsylvania and New Jersey. But besides that, just figuring out how to increase seed and seedlings that will be available for planting is going to be a really big challenge. If you look at current production across the eastern U.S. or across the United States, there are some 1.2 billion seedlings planted on 2.5 million acres. 82% of that is in the South. 75% is bare rootstock, and I'm sure you guys can guess what most of those seedlings are. They're mostly loblolly pine. They're mostly planted for commercial timber and pulp production. This is a picture of Penn Nursery in central Pennsylvania, and them growing some of our chestnuts. Certainly not 1.2 billion. So of those 1.2 billion seedlings that were planted, only 5% is hardwood, and 67% of all those hardwoods produced are oaks. So there's a huge lack of hardwood. I think both knowledge and production pipeline across the nursery industry that Chestnut's going to have to work to get into, both in terms of learning how to produce it. Right now, with all of our nursery partners, we're getting about 40% survival of the seed that we send to them. So if I have 100 seed, I'm only getting 40 of them in seedlings, and that's pretty rough when it takes us a lot of time to produce those and harvest them and get them into these nursery beds. So there's a lot of work to improve the pipeline of harvesting the seed, getting them planted, and getting the seedlings out to then plant in the field. So that leads us to, how are we adapting to climate change? We've done a lot of modeling. This is a recent paper done by Paul Noah, who is a student at Duke, looking at where suitable habitat is going to be between 2010 and 2100. And you can see, here northern Maine is really lighting up. The Adirondacks are lighting up, places where chestnuts do not currently grow, and where we don't see any of them is not part of the native range, are going to start being suitable, and we'll start losing even more that southwestern part of the range. So that's another aspect that we need to figure into what we're doing when we plant. And then we have to figure out, well, how much do we plant? So looking at these models of suitable habitat, there's an estimated 100 million acres of suitable habitat where we could plant American chestnut, and we're not planting 100 million acres. So how much do we plant? Let's say that we want to plant 1% of that suitable habitat. That would be 1 million acres. How long is it going to take us to plant 1 million acres? Let's say we can produce 2 million trees a year. We plant 500 trees per acre. That'll take us 250 years to plant 1 million acres. Let's say we reduce that, we go closer to that Rogstad and Pelican, that New Garden estimate. We can produce 2 million trees a year. We plant at 300 trees per acre. It'll still take us 165 years to plant 1 million acres, and that's just to plant it. So you have several hurdles. It's one of scale. I think it's certainly something that we can overcome. First, we have to figure out how to produce 2 million trees a year. We think we can do that in the next 10 or 15 years that we can get to that kind of production. Then you've got to figure out how to get them into the ground and get them to live. And then you just got to keep doing that over and over again. And that's that. We are excited that kind of challenge is looking upon us, but we also want to be realistic in terms of how long restoration is really going to take with a species that has been all but extirpated from its native range. And, tree planting is hard, but survival is even harder. You can plant trees in the ground, getting them to live with all of the things that they're facing. Deer are probably the hardest thing, the first biggest hurdle you got to overcome. Then you have all the invasive plants, mile-minute bittersweet, all these things that want to take over where you plant those trees. Other diseases and pests are out there. Phytophthora cinnamomii, we can save that for another talk. There's another critter out there, granulate ambrosia beetle, spotted lanternfly. Luckily, I'm going to knock on wood. It doesn't appear to be a huge issue on chestnut, but who knows what's out there ready to attack these populations. So it's not really just getting the trees in the ground, it's getting them to survive, regenerate, and repopulating areas. That's going to be really the biggest challenge. I am hopeful. I am optimistic. I really think that we have the tools, and working with all kinds of people, with the Forest Service, NRCS, local forestry agencies, NGOs, and private citizens, I really do think we can overcome all of those hurdles and get to a point. I love this picture on the right. This is my colleague Kendra with the Berlin tree in Vermont, and I call this the Kendra pose. Whenever we see a big tree, we do the Kendra pose. And we think that we'll be able to see this, maybe not within our lifetime. I don't know that within my lifetime I'll be able to see an 80-year-old tree that large and be able to hug it. But I think within my kids or grandkids, we'll see really huge advances in American chestnut restoration. It's going to be primarily due to volunteer involvement. I see that my time is getting limited, so and I want to leave time for questions. I want to mention that while Darling 58, this tree that we hope will be deregulated by APHIS within the next year or two, there's still other types of disease-resistant American chestnut in the pipeline. Darling 58 will not be the final quote unquote product in the landscape. It's great. It does still get the blight. It does not have phytophthora resistance. There are other things that are being worked on. There's something called Darwin that has a wound-inducible promoter that also has the oxogene that ESF is working on, that we're working on with them to diversify. We want to do more incorporation of breeding material with resistance from large surviving Americans and Asiatic resistance. And we also want to start working on, we've already started working on CRISPR and other gene editing technologies, and seeing if that can't be incorporated for increasing populations in the landscape. So this is a very long-term program. We're looking at decades, if not centuries, into the future, and it allows us to leverage new technologies that come along over time, and hopefully follow the template that we've already been able to succeed with in terms of marrying all these different technologies with the various partners and people working on it. So with that, I'll leave a wistful quote. I wish I could come back in 100 years and see my trees, and I think we all do. This is something that we do not necessarily for ourselves, but for generations to come, so that they can enjoy the fruits of our labor. With that, I will feel free to email me, or if you just look up the Chestnut Lady at Penn State, you'll probably find me. But I'm sff3@psu.edu, and hopefully we have some time for questions.